hope you had a great break. It is good to see you all back. Remember that you have the clinical assignment due today by 5. Some of you guys have already emailed it to me, and some of you guys have turned it in up here, so remember that. Hopefully, if you emailed it to me earlier, I replied to you. It seemed like a bunch of them came in the middle of when some of my DSEM students were also emailing stuff to me, so I got a little bit confused about student emails for a while, but I think I got it worked out now. And all of the other assignments for the rest of the semester are posted. So remember that you've got an assignment with primary lit due next Monday. There are a bunch of papers that you can choose from. I might add some more, might not. I don't know. We'll see if I find some more or not. But there's 15 to choose from, so it's not like you have some shortage. So just be aware that all that stuff is upcoming. Today, I'm going to be talking about hypersensitivity. I am not going to make it all the way through all of this hypersensitivity stuff today. Fortunately, I actually gave myself 2 days for it, so got plenty of time on Wednesday. Exactly how I organize some of the things on Wednesday will depend on exactly how far I get today, because I can imagine a couple different ways of organizing some of the upcoming material. So we will see where things go today. So you can see that this is listed as hypersensitivity. Usually, when I think about hypersensitivity, or when we think about hypersensitivity, immunologists are really using this as a fancy word for allergy. And so you can see another hypersensitivity image here. What you will notice as we talk about hypersensitivity is that there are a bunch of ways that it goes in common, or has some things in common with autoimmunity, like we talked about before. We are later going to be talking about transplantation, and there's also some big ways that this has some things in common with transplantation. If I were going to super oversimplify things, I would say that in the cases of hypersensitivity and autoimmunity, certainly transplantation, yes, but not as obviously. The problem is a problem of too much immunity. We're on the too much Goldilocks side compared to other situations where the problem is on the too little side. There are times, in fact, this happened a bunch of times in some of the slides I was doing, particularly right before break, where sometimes there'll be examples that are really hypersensitivity examples that'll show up with the autoimmunity stuff, or sometimes they can be a little wonky in terms of exactly where the line is between the two, because they do have a bunch in common in terms of sort of them both being on that Goldilocks too much side. Officially, autoimmune disease or autoimmunity is when you are having sort of an excess or inappropriate immune response to self, while hypersensitivity is when you are having an excess or too much immune response to an innocuous substance, so something harmless that you shouldn't be making a big immune response to. So if I make an immune response to insulin, that is an autoimmune response because that's a self protein. If I make an immune response to pollen, I'm not a tree, so that's not a self thing, but it's a harmless thing that I shouldn't be trying to make some kind of response to. So that's really officially where the definition lies. Some of them get a little weird. One example that I probably won't get to until Wednesday involves a innocuous substance chemically modifying a self protein. So is that exactly innocuous or is that self? Some of the lines get a little weird, but that's where the official definitions are. And the big thing that we should be thinking about is that we're going to see some common mechanisms between some of the different types of diseases that we are seeing. Right now, we're talking about hypersensitivity or allergy, and we are going to see that there are 4 different types of hypersensitivity reactions. So we're going to be learning about the 4 different types of hypersensitivity reactions, kind of what those allergies look like, a bunch of details of them. I told you a bit before when we were talking about autoimmunity that some of our autoimmune diseases will actually parallel some of those same types of hypersensitivity responses. When we talk about transplantation later on, it's going to, again, be kind of the same kinds of mechanisms. So if you can understand these 4 mechanisms really well, then you're going to be kind of understanding the same things that we see many times over and over. The top part of this slide is from an older edition of your textbook. The bottom is the hypersensitivity table or figure from your textbook showing the 4 different types, and we'll be walking through these 4 types of hypersensitivity today. This is just another way of looking at those 4 hypersensitivity reactions. So when we are distinguishing between the hypersensitivity reactions, we're going to care a lot about which immune cells or proteins are playing a role. Notice that for 3 of the 4 hypersensitivity reactions, we have something that's related to an antibody. So in one case, it's an IgE antibody. In one case, it's an IgG or IgM antibody. That's actually true. I could also say for a type 3 that an IgG or IgM is true there. So we can't just say antibodies, because it turns out 3 of them seem to be antibodies, and you can see one of them is T cells. But there are big differences in terms of which types of antibodies. I'll also say that we're going to care a lot about things like mast cells with type 1. When we're thinking about types 2 and 3, the big thing that's going to be a differentiating factor is going to be something about the antigen. Is the antigen on the surface of a cell, like this green triangle? Or is the antigen a free-floating molecule, like this green oval? And so that's going to be a big part of our distinguishing factor. So we can think about which types of immune proteins or cells are really important, what kind of antigen is important, and then we'll also think a little bit about kind of how that happens and what that really looks like. Most of what I tell you today, or at least where we're going to start, is with the type 1 hypersensitivity reaction. The naming on the type 1 hypersensitivity reaction reminds me of one other thing that I should have said on the previous slide. One other thing that can vary between some of our hypersensitivity reactions is the timing from when you see the antigen to when you have symptoms. Note that we're going to put a little asterisk on that. You'll see what's going on with that asterisk as we go on. But there does seem to be a special timing with 2 of the 4 hypersensitivity reactions. And so a type 1 hypersensitivity reaction is also known as an immediate type hypersensitivity reaction because the timing is so important. So usually when we have an immediate type hypersensitivity reaction, we're seeing something that you make a reaction to sometimes within 10 to 15 minutes following exposure to that antigen. And we can think about that 10 to 15 minute business moving forward. A lot of times, and I myself am super guilty of this, a lot of times people, when they say allergies, they actually usually mean type 1 hypersensitivity. So if you try to imagine allergies in your head, you're probably actually imagining a type 1 as opposed to the others. Though I'll for hypersensitivity reactions can be an allergy type of response. You can see an example of what happens with that type 1 hypersensitivity reaction here, and you've got some information in a table here as well. So what you can notice is that we are going to have antibodies that are going to be really key here, and those antibodies are antibodies of the IgE isotype. Those IgE antibodies are going to bind to mast cells, specifically to the Fc epsilon receptor on mast cells, and those mast cells are going to then release their contents. And so usually when I think of the mediators of type 1 hypersensitivity, I think of both IgE and I think of mast cells as being the key mediators. And type 1 hypersensitivity can be a lot of different things. It can include anaphylactic shock that you may be aware of, and I'll say some more about that later. It also can involve things like hay fever, so my sad pollen allergy, asthma, hives, food allergy, and eczema, which is a skin type of thing. There are also types of dermatitis that can be in here as well. And this shows some details of what might happen with an immediate type hypersensitivity reaction. And this gets a little bit at the asterisk that I mentioned before about the immediate type hypersensitivity reaction. We can imagine this person who is being exposed to pollen. You might imagine, for example, this is me. I grew up in super far north, upstate New York, and then I went to college in North Carolina. There was different pollen when I went to college. So you could imagine that perhaps there was some time where I was first exposed to North Carolina pollen, this new first exposure to pollen when I was like 18. At some point, those pollen allergens got into antigen-presenting cells, probably somewhere in my lung. And those antigen-presenting cells presented antigen to T cells. Those T cells were Th2 cells. They made the B cells start making IgE, kind of gave me that whole Th2 type response, IgE response. And then I made a whole bunch of IgE that responded to that pollen. That IgE started floating around my blood, and the IgE was immediately captured by some mast cells that have an Fc epsilon receptor on their surface. Okay? And so we get to this point where there are these mast cells that almost have like a pollen receptor on them. They actually have Fc receptors that happen to have caught the pollen-specific antibody, but it almost makes it like I have pollen-specific mast cells. So imagine this situation. Imagine that, here we go. Imagine this is in April of my senior year of high school when I went to visit my college in North Carolina and decided that I was going to go there. Had this happen. How do I feel at this situation? What is going on? What's happening to me during these events? Yeah, Michaela. No, I'm not. Yeah. I'm totally fine. Nothing has happened to me at this point. I'm totally, completely fine the first time I see the pollen. So I'm like, this college is great. I'm going to go here. I go. And then during my freshman year, I see the same pollen again. Second time I see the pollen now. Now, my body already has some of these mast cells with IgE antibody. At the beginning, those B cells hadn't class switched. Remember, B cell starts with IgM. It had to do the class switch. And the class switch only happened because the T cell had to be a Th2. Like all this business had to happen before I could get IgE all over my mast cells. But now, freshman year, I already got some mast cells that have IgE on them. And now when I see that pollen the second time, now how do I feel, Michaela? Now I start to feel awful, because what's going to happen is that antigen is going to bind to those antibodies and cross-link them. We're going to bring some of those receptors on the surface of the mast cell close to each other. And now that mast cell, which has all of these granules in it, kind of ready to go, is going to release all of those granules in order to sort of act against the microbe. And that action is going to be pretty immediate. This is what the mast cell kind of looks like floating around my body that second time. That mast cell doesn't need to undergo cell division to do this. It doesn't need to do transcription. It doesn't need to do translation. It's got all these black granules ready to go. And so it can release them immediately upon being triggered. It really does not have to do anything. And so it can be 10 to 15 minutes where I can have those symptoms. And so I have this immediate type hypersensitivity the second time, and later times I see the antigen. The first time, nothing happens to me. But the second time, and all the rest of the times, now we start to see this type of response. And specifically, those mast cells are making a bunch of different products that were in these granules that are being released. And some of those mast cell products are shown here. What you can see is that some of those things are enzymes. Those enzymes can be important in terms of maybe changing connective tissue. It's especially important for things like wound healing, but you could also imagine that if I had a parasite, those enzymes could help be killing the parasite. We're going to see some other mediators, like my unfavorite friend histamine, whose structure is shown in the upper right, that is able to both kill parasites, but also change some things about circulation and cause muscle contraction. We're going to see the production of some cytokines. We're going to see some production of a whole bunch of lipids, all of which are coming from the mast cell to kind of induce some inflammation, to attract a number of other cell types, to get them to come to that location, all of those things. One of the things that I just point out here is one of those lipids that's made is arachidonic acid. You can see that arachidonic acid is listed, but we also have these other lipid molecules, prostaglandin D2 and leukotriene C4. You can see that there are enzymes that are involved in producing them. One of them you can see is cyclooxygenase, or COX. That's what some of your aspirins hit, is COX. And so all of these things may be produced by the mast cell. They're in those granules that are automatically getting released. And these products will have a number of different physiologic effects. So some of them you can see, like those enzymes, will lead to some tissue damage. That might be good for dealing with a parasite if you had a parasite. It's not great to just damage your tissues. Some of them are going to lead to things like vasodilation and changes in vascular permeability. Those were the same things that we saw with inflammation earlier in the semester. And so we're going to see things like histamine and some of those lipids giving us vasodilation and changes in the vasculature. We also are going to see some changes in terms of muscle contraction, which will eventually lead to things like bronchoconstriction or intestinal hypermobility, which AKA your intestine wants all the things to go out. Your intestine hyper, too much, mobility. We're moving your intestine too fast. We're trying to get everything out. And both of those really have to do with kind of how smooth muscle is moving. As well as those cytokines potentially leading to inflammation. And so if we happen to see some of this acting at the respiratory tract, you can see we're going to have that smooth muscle contraction that I mentioned before, and the smooth muscle in this case might be the throat or airways. That might make it hard for you to swallow, hard for you to breathe, and might make you wheeze. If it's the same kind of smooth muscle contraction, but it's happening in the GI tract, we might have things like stomach cramps, vomiting, diarrhea, all that fun stuff. So all of these types of symptoms are all happening from those same products coming out of the mast cell. Early on, we tend to see a lot of the things that are happening be related to kind of more changes to the vasculature, changes to the blood vessel, things like that muscle contraction, blood vessel permeability. While later on, cytokines will actually kind of change what's going on in the response, and we may see more long-term chronic types of effects, in addition to the more kind of structural effects that we saw in the acute early stages. And you can see this here. So at the beginning, we're mostly going to see those histamines and those lipid mediators causing anatomical changes. And so here you can see like a little bug bite happening because we had a little bit of leakage of fluid into that area, a little bit of swelling, a little bit of changes to the blood vessel. And then as time goes on, we get all this inflammation happening in this same region because now we're actually seeing much more cytokine-driven inflammation coming to that same area. Some people seem to be predisposed towards making these types of type 1 hypersensitivity responses. And I mentioned this a bit before as well. So based on either on details of genetics and other things like perhaps some environmental factors, some people tend to make a lot of these Th2 responses and IgE and will make type 1 hypersensitivity responses to just about anything. These individuals are known as atopic individuals or people who have atopy, where they will often have things like pollen allergies, allergic rhinitis, rashes on the skin, dermatitis, asthma, all of those things. I am like the poster child for a person with atopy. And so sometimes you might even be genetically predisposed towards making this type of response more frequently than other people. If we actually look at the genes that are involved, you can see that a lot of those genes tend to be, say, if we look at the ones that are involved with atopic dermatitis, some of them have to do with the barrier like the skin, and some of them have to do with other aspects of immune regulation. If we look at genes involved in asthma, oh my gosh, immune regulation types of things, and one that's involved in the airways. And so these types of things might predispose you towards having that type 1 hypersensitivity in different locations. These are some of the things that are thought about as being major type 1 hypersensitivity antigens. And so what you can notice here is that we've got ryegrass, ragweed, timothy grass, birch tree, penicillin, bee venom, wasp venom, cockroaches, dust mites, milk, eggs, seafood, nuts, animal hair, latex, mold spores. We got some nickel. So you can see a lot of really diverse things are being listed here. And if you imagine a person having an allergy to latex, and you imagine a person who has a peanut allergy, do those two things look the same? Or a person who has, and compare that to my being allergic to all the trees. Does that look the same as that latex allergy and that nut allergy? Or if someone was allergic to a cat. So if you actually think about this, you might say, I don't believe you that those are all the exact same response. I don't believe you that we're actually seeing this same kind of straightforward, simple mechanism that you just mentioned, leading to all of these types of things that we are seeing here. But in reality, we are actually seeing the same things happening in each of these situations. The difference is that the question is, where anatomically is that same thing happening? And so a type 1 hypersensitivity reaction always works like I just showed you, and you'll see this example happening over and over again. But it can actually look really different depending on what anatomic location it's happening in. And so sometimes you can see that type 1 hypersensitivity reaction happening in the bloodstream, and that's really severe and bad. That's anaphylaxis. Sometimes it's happening in the skin. Sometimes it's happening in the eyes, nose. Sometimes it's happening in the respiratory tract. Sometimes it might happen in the GI tract. And so we're going to see the same big picture immunological process happening in every one of these cases, but then the way that that's going to sort of mess with the particular anatomic organ is going to deal with kind of the anatomy and physiology of that organ. And so we'll see a lot of this in these examples. So one example we might see is known as the wheel and flare reaction, which is really just what happens when you see type 1 hypersensitivity antigen in the skin. So this means the first time, well, the first and second time that you're seeing this antigen, you're seeing it in the skin. So it's unlikely that, I don't know, I was going to just use an example that's actually a really bad example. So latex allergy, you're probably going to be seeing that latex in the skin. You're not going to be eating latex, right? And so you're not going to be seeing it in the GI tract. And so we can imagine, this is a situation where we see that antigen in the skin. We activate mast cells that live specifically in the skin. And the mast cells specifically act on blood vessels in the skin in order to make them more leaky, make them bigger in size, and give us these sort of traditional looking little swellings that you see in the skin. Oftentimes when you have a bug bite and it's a little bit swollen up and itches, a lot of that can be a type 1 hypersensitivity reaction to mosquito saliva. And so basically you're getting sort of a little bit of this inflammatory response because you activated some mast cells in the skin that had IgE coating them. And you did that when you encountered the antigen on your skin. You could also encounter the antigen in the respiratory tract by breathing it in. That might be pollen. That also might be dust mites. This is an example of a dust mite. I think for a long time I didn't exactly know what the deal was with like dust mites and dust allergies. So dust mites are these little tiny microscopic arthropods that are actually eating the dust. It's not the dust itself you're allergic to, it's the little tiny arthropod. If you really want to get grossed out, you can learn about face mites. There are actually little arthropods like this all over your face all the time. Anyway, dust mites, these little tiny arthropods, have a protease antigen that is really the problem. So it's not the dust itself, it's these little tiny unseeable mites that are eating dust that have an antigen on them. That's the thing you're allergic to. In any case, you would be breathing in this antigen. And so now the antigen is not entering and seeing mast cells in the skin. Instead, we're seeing mast cells in the respiratory tract. And again, we're going to influence blood vessels in the respiratory tract. But the respiratory tract also contains a bunch of smooth muscle. And that smooth muscle is going to start to contract to constrict the airway, leading to potentially difficulty swallowing, breathing, or wheezing. We're also going to see a lot of sort of those contractions allowing you to try to expel the contents of the respiratory tract through coughing and sneezing. You'll also get a lot of mucus secretion in this particular location when you have mast cell activation. So it's the same process of you had class switch to IgE, you coated up your mast cells, now your mast cells got triggered. The difference is where were the mast cells that got triggered? Here it's in the respiratory tract, and that leads to coughing, sneezing, constriction of smooth muscles, all coming from those mast cell components. Same thing with food allergy. It's just that now the place where the mast cells are is in the GI tract. So here we can have an antigen that we ingest. It's now going to activate the mast cell in the GI tract. We're going to get inflammation in the GI tract, but we're also going to see a lot of muscle contraction in the GI tract. And that's going to lead to things like stomach cramps, vomiting. Some of that inflammation is going to lead to changes in fluid secretion, where we're going to potentially have more fluid going into the gut and diarrhea. We're going to start to potentially have expulsion of GI contents because of some of those muscle contractions. So again, it's the same thing. There was some IgE on the mast cells, and then the mast cells saw the antigen, and then we got some inflammation, and we got some changes to the blood vessels, and we got some changes to smooth muscle. But it's just now where those mast cells lived in terms of what that ends up looking like symptom-wise. And so this kind of gives you some of the details of a lot of different types of food allergies. Technically, you can actually have a lot of things working as food allergies, because if you think about it, some foods you have to like pick up and eat. So you might actually encounter the antigens partially in the skin, and so you might end up with rash symptoms. Some foods you're going to sort of be inhaling, some of as you're eating it, and so you might get some respiratory symptoms. And so sometimes you get, this is real, mom, I promise I really have this, that you get your mouth gets itchy with certain things, because you may have that first antigen coming into the first place we're seeing the antigen. So we can kind of see a lot of different types of ways that food could potentially trigger some kind of allergy. Sometimes people will ask questions about things like exercise-induced asthma, and I don't have a lot of good information for you about that. I apologize. It does show up here, and I can't answer any questions about it. And so here you can kind of see some of those details of food allergy. And I have some specific things I want to mention about food allergy, which is a little bit why I'm sort of showing you this. Oftentimes what might happen, something you might imagine happening, is that perhaps the first time you see that allergen, maybe it's through the skin, right? You make your Th2 response, you make some IgE. Is that IgE going to stay in the skin? Where do you think, so where does the antibody come from in the first place? Yeah, Grace. It comes from a B cell. It's secreted by a B cell. Where's the B cell? Maybe the skin, but maybe the blood, or maybe a secondary lymphoid organ. And where is the stuff it secretes going to go? It's going to circulate in the body. It's going to circulate through the blood. And so you're probably going to get IgE everywhere. So even if the first place you see the antigen isn't, say, the stomach, if you see it somewhere else, make an IgE response, and then suddenly eat it, you're going to have those mast cells in the GI tract who are going to get IgE on them because the IgE circulates throughout the body, and you're going to suddenly potentially have this problem. And so here you can see we see this allergen for the first time through the skin, but maybe sometime it later comes in the intestine, and now we're going to see this food allergy sort of happen. I don't have it in here. Okay, I'm telling you like a whole side story now that I don't have the slides for right now, but it's so interesting. So one of the reasons why this is sort of interesting, and one of the reasons why this has always been a little bit curious, is that we have learned that there are different immunological environments in some different parts of the body. And it turns out that one part of the body, well, a bunch, but one in particular that matters for this story, tends to have a lot of cytokines and a lot of other things that push T cells towards a Treg phenotype. So if a T cell sees its antigen in that part of the body, it might get pushed towards Treg. Okay? And that is the GI tract. So in fact, it's something, we have this process that we know as something called oral tolerance, where a lot of times if the first time you see an antigen is through the GI tract, you'll get tolerant to it. You'll make a Treg response because the GI tract is sort of biased towards making a response to being a tolerogenic response. Why does it make sense that if you first see an antigen through your GI tract, you might want to have tolerance, especially if there's no like inflammation going on? Yeah, Kyra. So you can eat food. Yeah, most of the things that you're seeing in your GI tract are food antigen. Like steak protein is an antigen. You don't want to it'll make a big response to that. And so the GI tract has evolved that if you see an antigen for the first time through the GI tract and there's not like crazy inflammation going on, like there might be in some kind of GI virus, that you're going to make Tregs and you're not going to make a response to it. And that leads to some questions about like, well, then what's up with food allergies? And like, how might that be related to food allergies? And one of the things that is hypothesized is that perhaps in at least some cases, people are being exposed to some of these antigens the first time, not through the GI tract, but through some other place in their body. So they're not making the first response be a Treg response. The first response they're making might be this Th2 IgE response. And so they might have allergy because the first time they saw it was through the skin or through the respiratory tract or something like that, when maybe what should have happened is that the first time they saw that antigen should have been through the intestine. So I'm going on this long diversion to tell you that one of the things that people are thinking about right now is the dramatic increase in the number of kids who have peanut allergies and the severity of peanut allergies. Like when I was a kid, like the idea that like you couldn't bring peanut butter sandwiches to school because there might be somebody in your class who like, no, people didn't have peanut allergies that severe, and like not very many people had them, and like they really increased pretty dramatically in severity. And one of the hypotheses and one of the things that people are thinking about, and there's some data on this, is that in that time there's been a big change in lotions that are used for babies, then they now have peanut antigen in them. They have a peanut oil in the lotion. And so babies are now often seeing peanut antigen for the first time through the skin instead of through the GI tract, and are making this type of allergic type of response, so that then the next time they see peanut, they're having the allergic response, the type 1 hypersensitivity, instead of going straight to the tolerogenic response to peanuts. And so there have been sort of people who are, there's ideas about like feeding babies peanut containing foods really early and things like that, with the idea of trying to kind of counteract this, or thinking about whether there's peanut oils in baby lotion. So like little things like that could actually end up playing a big role in some of this stuff. Yeah, Emma. Yeah.
One of my like really good friends is severely allergic to like a million different foods, like peanuts, eggs, shellfish, sesame, like literally everything. So is that just because she's more prone to being hypersensitive, or is there something with the antigen that it looks similar in a way that your body's just overreactive?
I could imagine a bunch of different things going on there. I could imagine a general immune regulation issue. I could imagine, not setting up a good tolerogenic environment in the GI tract. I can imagine a lot of things. It's really hard to say. So I've mentioned a bunch of different ways that we can see mast cells getting triggered in different anatomic locations, and that leading to different types of allergy symptoms. The one anatomic location I didn't really mention is what happens if that antigen is actually in the bloodstream. And if we have our antigen in the bloodstream, we're going to be impacting mast cells everywhere, instead of in just kind of the one location. And so we're going to be changing blood vessel diameter and permeability everywhere. We are going to be making smooth muscle contraction happen everywhere. And this, of course, is going to be an immediate type of thing after our exposure to the antigen. And so in this case, we're often going to see issues with the heart and vascular system and the respiratory tract and the GI tract all at once. We usually see some hives, swelling throughout the body, and this is in fact often can be pretty lethal for our patient. And so again, this is actually in fact the same thing that's happening when I'm having a pollen allergy. It's just that when I'm having my pollen allergy problem, it's mast cells in my lungs, as opposed to here, it's mast cells in my whole body that are reacting. So this also is a little bit of an answer to your question, Emma, or at least hits at some of your question as well. And there's sort of this question of like, okay, why exactly why have I evolved to respond to pollen? I would really like it if I did not respond to pollen. When I used to teach this class in the spring, I used to give during these lectures, I would be giving an in-person demo of pollen allergy, because it would be in the pollen time. I'd really be happy if I didn't have this. And so there's sort of also been these questions in terms of why has this evolved? And one thing that we have noticed is that many of the really common allergens are relatively similar to antigens on different types of parasites. And so there are some structural similarities between, say, some parts of cat dander and helminth antigen, or pollen and helminth antigen, or cockroaches and helminth antigens. Again, you can imagine, if the dust mite is actually that little tiny arthropod, you can imagine it having some similarities to a little tiny parasite. And so I think there's been this idea that perhaps some of these things are really kind of cross-reactivity types of issues, though there have been some other arguments that people have made. And so the idea with this is that, we evolved this response to get rid of parasites. We evolved this response to get rid of helminths. Now I have no helminths, but I have this cross reactivity with this pollen antigen that's basically, triggering the T cell that would have been triggered by the helminth if I ever had helminth infection. And you can do the biochemical analysis, and that makes a lot of sense. But there is one kind of thing about this that doesn't totally make sense. If you think about a lot of the symptoms we saw with a lot of the different types of reactions, and particularly I'll say, think about the GI tract symptoms and the respiratory tract symptoms. When you think about those symptoms, and you were gonna oversimplify them and explain them to a little kid or something, how would you describe what those symptoms all have in common? What do sneezing and diarrhea have in common? Yeah. You're getting stuff out, right? You're trying to take whatever's in your body and get rid of it. And you're trying to do it immediately. Remember that a type 1 hypersensitivity response is a very, very fast response, 10 to 15 minutes. You're trying to get stuff out real fast. Parasitic worms tend to cause problems over many years. And so sort of this question of do you really need a in 10-minute response to expel the worm in 10 minutes? Does that really make sense as how you would have evolved a response to it? You could be well, maybe you just really don't want it to get in the first place. It seems a little weird, but you can kind of argue it. But there's always been this question of why so immediate? Why did we make this response that is so immediate? And there is this counter hypothesis that actually also notes that there are quite a few cross reactivities to things like venoms here as well. And so this may actually be a way to expel venom if something bit you. Because in that case, you do want to get that out ASAP, really immediately. One other thing I'll just mention about the type 1 hypersensitivity reaction. So let's imagine that you are hanging out, you go this weekend to the city and you ride around in some horse-drawn carriages. Okay? You go with your friend and your friend's like, oh my gosh, I'm so excited. I've never been in contact with a horse before. I want to see a horse and go to the horse-drawn carriage. Your friend goes in the horse-drawn carriage and starts sneezing right away. What do you know about your friend's claim about whether or not they've seen horses before? Yeah. They're lying. Because you only see, remember, that type 1 hypersensitivity response the second and later times you've seen the antigen. It doesn't happen the first time you've seen the antigen. So remember that a really key thing here is going to be the IgE and the mast cells. I point that out because this is going to contrast with some other things. And when we think about what kind of medications might work here, medications that hit T cells, not helpful. T cells aren't the problem. I can give you all the T cell medications you want. That's not going to help you. So remember that this is IgE and mast cells. Remember that it's 10 to 15 minutes, but it's the second time or later. And so you will see on old exams all these little stories, and they're always has this ever happened to this person before? Has this person ever encountered this random antigen before? So remember, that's a thing you should be looking for here. So we're now going to switch gears into thinking about type 2 hypersensitivity. I remember as a student being a little confused about some aspects of type 2 versus type 3. One thing that is really important about type 2 hypersensitivity is that we've got antibodies, and again, it's an IgG or IgM antibody. But I will tell you that type 3 is also going to be an antibody that's IgG or IgM. So you can't just remember IgG or IgM antibody for type 2. You got to remember one other thing to help you understand how this is different than what we're going to see in type 3. And the thing that's really important with a type 2 hypersensitivity reaction is that the antibody is against a cell surface antigen. So the antigen is on some kind of cell. So here you can see the antigen is on the surface of a cell in this little picture. And the reason why that is important is that what's going to happen in a type 2 hypersensitivity response is that we're going to hurt this cell. We're going to damage the cell that has the antigen on its surface. And so this damage can happen in a few ways. It could be complement. It could be ADCC, which remember is NK cell activation. Could be a few things, but the key thing is that there was a cell with the antigen on its surface, and we destroyed that cell in a type 2. And that happened because IgG or IgM bound to the antigen that was on the surface of the cell. So one of the really famous types of a type 2 hypersensitivity reaction is related to red blood cells. And so in order to tell you all about this particular situation, I need to tell you a little bit about red blood cells and blood group antigens. So you are probably aware that people have a blood type, either type A, type B, AB, or O blood types. This is actually based on the presence of some carbohydrates on the surface of their red blood cells. So people who have A have this turquoisey hexagon and acetylgalactosamine. People who have B have this blue galactose hexagon. People who have O have no hexagon there. When we think about the genetics of this, it's actually about what enzyme do you have to add the carbohydrate to the end of this chain. And so these are all different carbohydrates on the surface of red blood cells. You can also see there's another antigen that some people have called Rh antigen. They're Rh positive. Other people don't have Rh and are Rh negative. So when we think about blood types and blood type antigens, there's this other piece that we think about. And I remember learning this in like high school biology, and I was like, I memorized it. Yay. And then I saw it in an immunology class, and I was like, that violates like all the immunology you just taught me. Because they didn't tell me like one other piece. So I want to make sure I tell you like the whole story and don't just tell you the confusing stuff. So some people have the A antigen, which is this one that's shown here, this carbohydrate on the surface of their red blood cells. You can imagine that at some point in that person, there was a B cell that made by VDJ recombination a heavy and a light chain to make antibodies against the A antigen, right? What happened to that B cell? Yeah, Josh. It probably got killed or otherwise dealt with by central tolerance mechanisms. So we do not have that B cell, so we are not going to have any antibodies against A. What about a B cell that did VDJ and made a receptor that bound to the B carbohydrate? Is it possible for that B cell to exist in that person who has type A blood? So I see lots of yes head shakes. Why we have yes head shakes? Yeah, Emma.
Because the antibodies targeting anaphylaxis, that's foreign to the person with type A blood.
Right. Well, also I want you to remember, like VDJ is a random process, so we're kind of making one of everything. So yeah, we're gonna make that one amongst all the other ones. But how many of that B cell are we gonna have? Kind of just said it in my previous sentence. We're gonna have like one of that B cell. B cell's probably not gonna like be activated, right? And like become a plasma cell and go through all of that B cell activation stuff we talked about. Except that it is. Because I think that one thing that sometimes people are like, but okay, so you're gonna what I tell you is that if you have type A blood, you have lots of antibodies against B. And my question is, well, then how did I get the how did I activate the B cell? Like I can understand why I have the B cell, but if I don't have this antigen in my body, how did I activate the B cell? It turns out these carbohydrates are on the surface of all of the microbes in your GI tract, and so basically, if these B cells exist, they get activated and you make boatloads of antibodies. And so in a person who has type A blood, both the type A and the type B cells got made during B cell development. The A1 got dealt with in central tolerance because it was a self-antigen. The B1 got sort of primed up by those microbes in the GI tract, and so we have lots of antibodies against B in this person's blood. Same thing if we have a person who has just the B antigen on the cells. The B cell got dealt with by central tolerance. The A antigen specific B cell got lots of priming, so we're making boatloads of antibodies. If we're AB, both B cells got made again, but they both got dealt with by central tolerance, and so we don't make any of these antibodies. If we have type O, then none of these things were on the surface. None of them were self-antigen. We didn't kill either B cell, and we made boatloads of both antibodies. So this is kind of the general way that blood types work. What can happen, unfortunately, is this. But I guess the point to first make is all of these things, these carbohydrates and the Rh antigens, are on the surface of red blood cells. And so they are cell surface antigens. So remember, I told you that with a type 2 hypersensitivity response, we have an antigen on the surface of some kind of cell. And so the most famous place where this is a problem is something called erythroblastosis fetalis, also known as hemolytic disease of the newborn. And so what can happen is we can have a mom who is pregnant with a baby that is Rh plus, while mom is Rh minus. So if mom is Rh minus, note that she probably has a B cell that responds to the Rh antigen. Rh, we don't really make a huge number of antibodies against. But when mom delivers this baby who's Rh plus, there's going to be a little bit of the baby's red blood cells that are going to go and be exposed to mom's circulation, like through the placenta, through delivery. And suddenly, the baby's Rh plus blood cells are going to come across that mom's B cell that binds to Rh and are going to stimulate it. And now mom is going to make tons of antibodies against Rh. Okay, whatever. If mom gets pregnant again with a second baby who's Rh plus, now these antibodies that have been made, these IgG antibodies that we've primed up, can go across the placenta and kill all baby's red blood cells. And so now we have this baby whose red blood cells are getting killed in utero. And so we have this baby who basically is born without red blood cells, is also has a bunch of has severe anemia among a bunch of other things. Happily, we do have a way to treat that now, and so this is actually not something we see super often at this point. Because what we can do is we can actually when we see, oh look, mom is Rh minus and she's pregnant with an Rh plus baby, we can actually treat her with monoclonal antibodies that bind to Rh so that this B cell never gets activated. So we never actually turn on her Rh specific B cell, and we never, activate it. We never get plasma cells. We never class switch to IgG. And so she never makes IgG to pass to that second baby. And so we'll just treat her with this antibody during any pregnancy that she has with an Rh positive fetus. But before this, we would see a lot of killing of those red blood cells because they had the cell surface antigen. So you can see this is that same thing. So, typically we might have mom who has this cell that's become an IgG producing cell. We're going to eventually have anemic babies. And so if we treat mom with this monoclonal antibody, it's going to not allow the B cell to become activated. So we never get class switched to this memory B cell, and mom never makes the detrimental antibody. Yes, I am absolutely not going into this slide in as much detail as I have in past years because there's a whole other issue that I feel like immunologists have suddenly gotten uncertain about that I don't want to touch right now. So yes, I am very much glossing over this in a way that I have not always used to. Another famous example of a type 2 hypersensitivity reaction is a penicillin allergy. I will point out, however, and this slide shows it, that penicillin can actually lead to all 4 types of hypersensitivity reactions. So if you have a penicillin allergy, it could be a type 1, type 2, type 3, or type 4. I'm going to tell you about a type 2 example, but there are other types. And with the type 2 type of penicillin allergy, we again end up with a type of anemia, again because we're going to be killing red blood cells who have a cell surface antigen on their surface that binds to antibodies that then leads to those red blood cells getting killed. And you can see penicillin in the middle. Penicillin has this very reactive bond. It is supposed to react with a bacterial protein to inactivate that bacterial protein. But in some people, penicillin will react with one of those carbohydrates on the surface of the red blood cell and attach itself to the red blood cell. And now this becomes a new antigen on the surface of the red blood cell. So we've got this thing that should be harmless, attacking a self-antigen and modifying it. And now we have this new antigen on the surface of our red blood cells. And those red blood cells can be killed by IgA or, So we can see here's the red blood cells with penicillin on the surface. Now we can have antibodies like IgMs or IgGs. And we can either destroy those red blood cells with complement, or we can phagocytose those red blood cells. Either way, we have not enough red blood cells, which leads to anemia. And so all of this is because of this cell surface antigen on the surface of the cell. On Wednesday, I will start with thinking about type 3 hypersensitivity, which is known as immune complex disease. And so I'm going to talk about that, and then I'm probably going to do some big picture immunotherapies and do transplantation on Friday, though there's a chance I may try to go into transplantation and do all the therapy stuff at the end on Friday. We'll see. So I will see you guys on Wednesday. Have a great next couple days.